Cell signaling is a very, very complex storyline. We have different ways that cells can release chemicals, showing you just this is the communication, this is the language of how cells interact. Autocrine is defined as when a cell is able to self-regulate by releasing a chemical compound that could bind to its own receptor. So the chemical signal, whether it be an ion or solute or ligand, it will bind to its self-receptor and elicit a response. Some cells have the ability to do that. You can also have signals that are paracrine. Prostaglandins, eicosanoids, it was one class of the lipids. Those chemicals can alert nearby cells that are adjacent to one kind of cell type. And these are usually related to inflammatory responses. Endocrine is when you have a dedicated gland in the body, the hypothalamus, the thyroid gland, the thymus gland, testes, ovaries, you name it. So any of these dedicated glands that have the ability to release a chemical, and when that chemical can only bind to a specific target cell. The first type of signaling mechanism are called metabotropic receptor signaling. This is also equivalent to what we call G protein receptors. The word G protein means it utilizes a nucleotide called GTP. Okay, so it's a G protein receptor, and this kind of signaling has a plot. Whenever you hear the word metabotropic or G protein, that just means that that chemical that binds to this receptor, that is a metabotropic receptor, is going to activate some kind of enzyme that is on the cell membrane. And it's not shown here. So imagine there's another enzyme right here. And this enzyme will then activate another signal inside our cell. So you have, this is the first signal, right? So imagine this is signal number one. First chemical is the first signal, right? So your cells are just chilling there. And this first signal, whether it be a hormone or neurotransmitter or a chemical or drug, the second signal comes along when this receptor is bound. It will activate some random enzyme down the membrane, and that enzyme will make another signal. We'll use this little red circle here as the second signal, or otherwise known as the second messenger, same thing. And that will then activate some series of enzyme. Going back to what I just said, phosphorylation with a kinase, right? Kinases are going to be an enzyme that will turn something on. So that story plot, what makes this system really complex is the second messenger could be several different signals. It could be something called cyclic AMP. It could be something called calcium. It could be something called DAG, but there are a variety of different kinds of sub-signals that can be. The second type of signals and receptors that our body utilizes are a lot more straightforward. So thank goodness these are called ionotropic. The word iono means it's ion-based. These ones are going to be when a chemical, so imagine this is going to be the first signal. It binds onto the receptor, and now the receptor is activated and the door opens up, and now this little purple circle can go through. Acetylcholine. ACH is the universal excitatory chemical that triggers every single muscle in your body to contract. So when you are eliciting a muscle contraction, your nerves have to release a go signal. That go signal is acetylcholine. And the way acetylcholine works is that it binds to an acetylcholine receptor. There are two classes. One is called nicotinic, and it binds the same thing as nicotine. That's why when you smoke or vape, the nicotine rush is real because it's all over your body. And when it binds to nicotine, it allows the ion channels of sodium, calcium to open up, so it begins to change the charge of your membrane. The third kind of signals and receptors you will come across are considered nuclear receptors. Nuclear refers to the fact that they are inside the cell membrane, and it targets directly onto the DNA. So it's pretty intense. And when this hormone, let's say, comes along, gets into a nuclear receptor, it can dock on and allow the binding of it directly into your genes. So the example was testosterone and estrogen. And that's why when you look at, going back to that example of bodybuilding, if you look at a female bodybuilder, you can see a lot of traits that are extremely male-like, and it directly affects gene expression.